his pea plants. During this time, there was actually a different thought of genetics, and we already know that Gregor Mendel is going to be known as the father of genetics. But before that, certainly it was understood where children come from. And observing a child and then observing the properties of that child and pointing back to the parents was something that could be easily understood. But during that time, there was a different hypothesis than what Mendel thought existed. The major hypothesis of the time was something called the blending hypothesis. And what the blending hypothesis states or assumes is this is a theory where the child or the progeny, the offspring, is an average or is a mixture of the two parents. It's like taking red and white paint, mixing them together and getting pink out of it, right? But if you think about mixing and matching those colors, like take that pink and then mix it with the lavender from the purple and white mixture, and do that many, many times, what's the result generations later? A pretty uniquely brown color that everything is, right? And someone said muddy. So the result of blending hypothesis would be over the course of many generations that humanity has certainly lived through. By this time, we'd all be identical. So we have to have discrete heritable units and not be a blend of our parents.
How did they explain gender with that?
They couldn't explain gender till the turn of the century, 50 so years later.
Well, it's interesting.
Now, what's very ironic is that when you study biology, there's two scientists that come up that are like the cornerstones of the class. This guy's one of them. Who's the other guy?
Newton. Double Felix guy.
I think Darwin, right? Now, the irony of this is that kind of a couple continents away, but probably about across the world, you have Darwin on his little ship. Do you remember the name of the ship?
The Beagle.
The Beagle.
Good bagel.
It's the HMS Beagle, not HMS Bagel. And the Mayflower is what carried the Pilgrims over. So we're getting a very intense history lesson today. But Darwin came up with this idea of natural selection, which was his mechanism of explaining his theory called evolution. And though he never used the term evolution, he always said like a survival of the fittest and this reproductive success that could be explained, or that could explain his theory of evolution. But Darwin's natural selection, that's basically means survival of the fittest, right? Good traits survive, bad traits just seemingly die out because they do not survive long enough to propagate and produce offspring. But Darwin couldn't put his finger on what was passed on. And across the world, ironically, you have this guy that is putting his finger on it. He knows what's being passed on. And for that reason, Mendel is considered to be the father of genetics. The irony, in addition to the irony we've already mentioned today, is that during his life he wasn't known for this at all. In fact, his research got quietly put away for many years until it was rediscovered and actually made sense. He came up with something called the particulate hypothesis, which combats, or you could say contradicts, the blending hypothesis. In this, Mendel's theory would be that one can inherit discrete characteristics or units from the parents. And we now know that those units are the chromosomes, which are carried in the gamete cell. So the egg and the sperm fuse, each haploid, together making the diploid zygote. That actually brings up an interesting point. If you look at the progress of biology over the 100-year span, you can actually see what Mendel's work started and what Watson and Crick's work ended. Let me show you. Approximately the year 1850, you have Mendel, who's making a discovery, and what he's basically doing is he is identifying a heritable unit. I mean, he comes up with this particular hypothesis. There is something being inherited, if you will, but he can't quite put his finger on what. In other words, these pea plants were giving their baby pea plants something, but he couldn't quite figure it out. And then around the turn of the century, and I'm pretty approximate on this date, but I feel like I'm pretty close, around 1905. So let's just say, for the sake of argument, around the year 1900, with the advancement of microscopic technology, there's a discovery of meiosis. Of course, meiosis is the mechanism by which gamete cells are produced, and much like you saw in the lab from the other day, the mini lab, you could observe the chromosomes being segregated or manipulated in that process. So, meiosis was the story of having chromosomes. So, now all of a sudden we know that chromosomes are made or put into gamete cells, and gamete cells fuse to make an offspring. So the suspicion would then become that heritable unit that Mendel had, or that he had mentioned, has got to be the chromosome. But now a new problem pops up, and that problem's pretty simple. Scientists ask the question of what's a chromosome. And for 50 years-ish, the debate lingers, essentially. And that debate, Rob, is which part of the chromosome is responsible for acting as this heritable unit. Chromosome, of course, a chromosome is made of chromatin, which of course, chromatin condensed down forms your chromosome. That chromatin is made of DNA and associated protein called histone protein. So the question then becomes, between 1900-ish or so to 1950-ish or so, well, which one is it? Is it the protein or the DNA? Now, out of everything we've discussed in this class, who has been the more involved? Who has been the doer of, about everything? We put the blame on a protein about every time. And so scientists had about the same mentality, and so they were pretty certain that proteins were the heritable unit. However, with the discoveries in discoveries in between 1900 and 1950, coupled with the final theories of Watson and Crick, it's determined that DNA is the responsible party. I like to show this because this shows the progress that a science will take. The identification that there's something, and then the working, and not just of one person, but of many people, to determine what that something is. And it's a process that's shared among generations of scientists. Some vocabulary that I think you need to know, and this is vocabulary that you could accredit to Gregor Mendel. The first, the term character. What a character is it's basically a distinct, heritable factor or feature. A good example of this would be flower color. So this is a property that you can observe. This is one of the properties that he observed with his little pea plants. And then he saw that this character could vary. So he called those traits. The traits were the variants of a single character. Focusing our efforts on flower color still, he observed that the flower color could either be purple or white. So he was observing the phenotypes of these flowers, and he observed that there was a characteristic, if you will, which was the color, and that there was different variations of this color. They could either be purple or white. Now, one lucky thing, I don't know if it's luck or if it's just maybe dumb luck, but Mendel had a choice, or his choice of pea plants worked perfectly, because for a lot of their traits, a lot of the pea plant traits are inherited in an either-or matter. One thing about human traits is that they very seldomly do this, which would make his discoveries with humans, or making the discoveries he made with humans, would have been impossible. So lucky him for choosing the pea plant, or stumbling onto it. What do I mean? I said the pea plant could either have purple or white flowers, correct? That's like black versus white. Did I say they could be a shade of purple? Like a lavender? A off-white purple? They were either purple or white. Humans and more advanced creatures seemingly have traits that vary along a gray scale instead of just a black or white scale. And that makes it much more complicated to make discoveries with those types of traits. And he also came up with this idea called true breeding. What true breeding means is that it is a variety that can produce offspring that are only of the same variety. So in other words, a true breeding purple flower could only produce purple flowers. So a progeny or offspring would only have purple flowers. So Adam, if you had a true breeding white pea plant, what could its babies only be?
White.
Okay, so that's what true breeding means. And I want to show you, and I'm going to and you can imagine that I'm going to show you very quickly, comparatively speaking, to the time he spent on this, but I'm going to show you his eureka moment, his experiment that maybe put the light bulb on in his head. And I'm going to try to question you maybe the way that he might have questioned himself. And then I'll go back with the proof that shows that his logic and his theory of genetics is valid. He took what's called a P generation. The P generation stands for a parental generation. It is the first living descendants in his sequences of generations. And what he took is he took a true breeding purple flowered plant. And he crossed it, so he mated it with a true breeding white flower plant. Now, being a novice with these pea plants and now just starting out, I think whatever this offspring is, so whatever this F1 generation is, it's completely up in the air because you know nothing about it. So I'll just tell you what he observed. Here's what he observed. All of the offspring were purple. And so now I think it would be a good time to introduce the words dominant versus recessive. I also think that there's a good question that you would ask yourself at this point if you were Mendel. What would the question be if you were to ask yourself, what happened to the white color? Where did the white color go? That's a valid question. More experimentation would need to be done. It seems as though, well, obviously it's absent. And these plants are as purple as these. If you were to use terms dominant versus recessive, which would you say appears more dominant in this case, the purple or the white? The purple seems maybe just a tad bit more dominant, whereas the white seems a little recessive. And if he produced 1,000 of these little plants right here, they are all what color? Purple. What did he allow them to do? Well, mate with each other or simply mate with themselves. So allow them to self-pollinate. And this would create an F2 generation. And this is where it gets interesting. And I think this is where, if you sit down and think about the math and spend some time with it, the eureka moment could exist.
Thank you.
All done? Essay 2?
Yes.
Okay, perfect. He observed 705 of these progeny were purple and 224 were white. Pretty interesting. Now, if you do some reduction here and you just think about the numbers, that works out to be approximately a 3-to-1 ratio. And I think, what question, if you were Mendel, would you ask at this point? Yeah, where? How did that white come back? Certainly, you could say that this white color is from here, but this was a 1-to-1 ratio. Why a 3-to-1? And so this is where you'd go back to your little study or your little library, and you'd sit down with some numbers and some logic for a while. And this is what I think you would find. Using the idea of dominant versus recessive, and knowing that they are true breeding, the purple flowered plant in this P1 generation had to have been capital P, capital P. The word we would use to describe that is homozygous dominant. Now, if purple is dominant to white, what would that make this white flower plant be? Or homozygous recessive, right? Now, you can do this Punnett square in your head, or for the sake of time, no, for just the sake of showing, I'll do it off to the side. Big P with little p, little p. And all of these progeny from this first cross are going to be what? Heterozygous, which means they're all going to be big P, little p. Now, does that explain why F1 was all purple?
Yes.
And then you allow them to do what? Self-pollinate. So I would have big P with little p and big P with little p. And so this cross would be something like this. Big P, big P, big P, little p, big P, little p, little p, little p. And so how many of those are going to be purple from this last cross?
3.
And then there would only be one that would be recessive. So a 3-to-1 phenotypic ratio, but a 1 to 2-to-1 genotypic ratio, and more on that later. Now, what I've done here in purple is shown you the data that he observed. The green would be the things that he derived, and this would take a while to do, I suspect. And then, of course, like any good scientist, what do you always do? Run more. You repeat, and then repeat again, correct? So let me show you what he was able to do after all those repetitions. The magic of science, I guess. All those numbers. Obviously, this would take a long time to just simply count, but you'll notice that he was able to achieve, using this data, about a 3-to-1. And it didn't really matter what he seemed to study. Now, lucky him, he was studying something very simple, like a pea plant. Had he been studying human genetics, like height or blood type, if there had been a test for blood type back in those days, he would have found it was much more complicated than the simple pea plant. But that explains why pea plants are pea plants and humans are humans. We would be more crazy of genetics than a pea plant. Right? Okay. Questions about Mendel or his logic? That was fun. So some words I need to describe. I just kind of threw them out a second ago, but I'll keep them going. The word genotype versus the word phenotype. We've heard these before, but I want to maybe tweak their definitions a little bit, or at least how we view them. The genotype does stand for the genetic makeup, the genetic complement of a creature. Okay, there it is. Whereas the phenotype would be the physical appearance.
Yeah.
And what you've been taught is that the genotype ultimately determines an individual's phenotype. And I would agree to that, for sure. But then I think of, well, let's think about twins real quick. I don't think anyone in here is a twin, are they? But you certainly know that this school is famous for having twins, correct? By the time a set of twins is like y'all's age, 17, 18-ish or so years old, they don't look exactly the same anymore. Sometimes one twin has longer hair, the other one has shorter hair, right? One may be taller, one may be shorter. They may in fact talk differently or just plain look different. Now when they were little babies though, they probably looked almost identical. Couldn't tell the difference, I suspect. As they grew up though, and you could visualize that as they get into their 60s and 70s, they're going to start to look vastly different. But they're genetically identical. So how could their phenotypes change over the course of time if their recipe, their genes, are the same? I think your environment has a big effect on it. So you also have to include the environment. And what geneticists are starting to learn and understand more is that your genotype certainly, without a doubt, does play a large part in your phenotype, which is your physical makeup, your biochemistry, and the inner workings of your body. But your genotype gives you maybe a set degrees of reaction. So that means that your phenotype, there is not exactly one specific phenotype that a person can display. They can have what's called a norm of reaction. For instance, there are some flowers that bloom pink or purple depending on the quality of light and the intensity of sunlight and the quality of sunlight and also the overall temperature. So they code for both colors. The colors only exist when it's a certain time. Okay, a couple other things real quickly. The word dominant versus the word recessive. Mendel is given credit for coming up with those words. He also came up with this idea of a locus. I want to show you a picture real quick. This picture really, I think, has the potential to help you see the differences between the last chapter and this chapter. This is a picture of homologous chromosomes. How do I know that? Because it says homologous chromosomes off to the side. Okay, perfect. Does that mean that these chromosomes are identical?
No.
No, but they're coding for the same stuff, right? So these two chromosomes, one would be from each parent. Agreed? Okay. If you think about this, this would explain, okay, the concept of alleles. This would have the purple flower gene, but also it has the white flower gene. You notice that when we talk about a creature, it's always big P, or big P, little p, or something like that, right? There's always two letters of the same type. Where are they getting each of those letters from? One from each parent. Now, what if one parent was dominant for purple and the other parent was dominant for purple? What would that progeny be? Only purple. Homozygous dominant, we say. So we're understanding that those two letters, those two alleles, each allele comes from a parent. Now, if you think about it, how many alleles could you have at a maximum? I think only two. Do you know how many alleles there are for blood types? There's three. IA, IB, and I. Those are the three alleles. But at best, how many can you have? Two. One from each parent. I suspect that my mom gave me IA, so A blood type, and then my dad gave me just a little I because my dad is a blood type O. That makes me have A blood type. And the B, I don't even have it all. Make sense? And then also, I think it could help us understand this picture a little bit. This is a slide, or a picture of a slide, that's going over independent assortment, which was a very important aspect of this last test. If you look at this diploid creature, there are two alleles for two different characters, the Y character and the R character. Agreed? Why are there two Ys and why are there two Rs in this diploid creature? One is from his mom and one is from his dad. Okay. Now, when he makes gametes, he's going to reduce his count by half, correct? How many chromosomes? Well, no, let's not do that. I don't want to play that game anymore. I would say that there are two traits that are being described here. Agreed? Now, he is giving one single Y to every sex cell and one single R. How many options are there for Y? And how many options are there for? So that would mean there's a total of how many different gametes he can produce? Because they are independently assorted. Let me show you another question that tackles the same thing. This question, though, was from the AP exam last year. A couple things about this type of a question. It's called a grid-in question. They had 5 on last year's exam. About 40% of all students that took the exam got 0 of these questions right. I think you guys could actually get this question right. And we haven't really even done a lot of the class. All these genes they said were linked or unlinked? Unlinked, right? Which means independent assortment is at work here, correct? So this guy, this plant, is diploid. And you're trying to make a gamete that is haploid. Okay. This plant is heterozygous for each of these 4 different characters. Agreed? What is the chance that this plant gives to its gamete, whether it be an egg or sperm, I do not know. What's the chance that it gives the capital W or the dominant for W? 1/2, okay. How about for, is everyone paying attention? How about for the X, the little X, what's the chance that it gives the little X? How about for this one? How about for this one? So it's 1/2 and 1/2 and 1/2 and 1/2, correct? And there are all unlinked. So this is 1/2 times 1/2 times 1/2 times 1/2, or 1/16. And what's amazing to me is that there are 40% of students that took biology and couldn't simply answer that.
Why is it 1/2 instead of 1/4?
Because you're choosing one, and there are 2 different W's. In other words, what's the chance of choosing a big W from a big and little W set? 1/2.
It's not out of
That's correct. It's out of just that set, that letter. Oh, and for your fun fact, there were 5 grading questions last year. Less than 5% of all students got all 5 of them correct. Not many. And the AP people worked it out in such a way that if you were getting a 5 on the exam, you almost had to get all 5 grading questions correct. It's kind of ironic. Last thing I want to talk to you about, then we'll be done, except for a little bit of a worksheet I kind of found, is I want to talk to you about this thing down here called a test cross. So, a Mendelian test cross. Putting yourself back in Mendel's shoes just one last time before we finish. Let's imagine that you spot in your little pea plant garden a purple flower plant. What is its genotype if it's purple flowered? You don't know. It could be big P, or it could be big P, little p, and hence the need to create something called a test cross. So here's how you do a test cross. You always take the unknown, which again, it's unknown because it could be homozygous dominant or heterozygous. You take this unknown plant and you cross it with something that is homozygous recessive. Good? Now doing that, there's one of 2 outcomes here. Let me show you the 2 outcomes. Let's assume that the pea plant had been homozygous dominant. Would have been big P crossed with little p. Tell me about all the progeny, all the babies would have been what? All would have been purple, which is something that you could simply see by looking at the plants. Or what if the original plant had been heterozygous? 50% of them would be what? Purple. 50% would be what? White. So what is a test cross? Well, basically it's the uncovery of what the genotype is of a dominant creature or dominant trait. So it is the breeding of a mystery individual, one in which you do not know the genetics, with a homozygous recessive, and the reason you do this is to determine the alleles of the mystery individual. In other words, was it big P, or big P, little p? Yay? Done. That was a lot of fun, I think. No? 1 person? 0 people. Well, I thought it was great.